Vladislav Strmiska from Mewery, who's going to talk about, cultivated pork and microalgae base.
Thank you very much. I'm happy for microphone. I left my voice yesterday in church. Okay. Hello, I'm Vlada. I'm a food scientist for my master's, and for my PhD, I'm a cellular biologist. Now I'm a cell butcher at Mewery. In Mewery, it is a company working on my cultivated pork on microalgae base, which technically means that we are combining two different organisms together. So we grow pork cells and microalgae cells at once. The reason why we use a microalgae, these are three major parts that we are using microalgae as a FBS replacement, as a live microcarrier for scaffolding to maintain a little 3D structure. And third reason is a growth factor production. These process to cut off the growth factors from third parties, FBS and scaffold, use just microalgae, which grows like for no money, literally for free. Like it would grow in dirty water if you wish. Costs the price down for 70%, as we know. We unveiled first prototype of our hybrid biomass, where we co-cultured porcine muscle cells together with the microalgae. It's easily predictable the culture, the percentage of each organism in the biomass. And the prototype, we just fried a little meatball of 75% of meat cells, porcine cells, and 25% for microalgae cells. It was not that big, but one family of four could survive on that for weeks, I believe. So I will introduce a little bit our cultivated process. So we basically isolate primary cells from umbilical cord of the pigs, so we don't cause any harm. We don't have to slaughter any animals. This is just basically waste over the birth of new piglets. Then is coming to microalgae. Microalgae can grow, and they grow separately to mammalian cells before. And we also maintain, of course, the primary cells from pork. We work mostly with primary cells. We are a non-GMO company. So at the end, once we have enough biomass from each, then we can consider what next to do with that, right? So then we have a scale-up process in a bioreactor, or a shaker flask, or any suspension, where you will just add both organisms at once, and they can easily grow together and maintain a little 3D structure. Then you have harvest, and you have a hybrid biomass. We call this hybrid biomass because it contains two organisms at once. This is just a comparison for you to know what's the size, what's the difference between microalgae cells and porcine cells. So mesenchymal stem cells, which we work with, are in a similar size, for example, as other kinds of microalgae. It very much depends what kind of microalgae you're working with, because the sizes are way different. So it also depends for what nature property you want to use the microalgae. Of course, the red one looks pretty nice, right? It could mimic myoglobin in a muscle, so you don't have to think as much about a green color. Yeah, this is for you just to show that how happily our cells can in 2D grow together with the microalgae. The red dots, it's autofluorescence chlorophyll of microalgae cells. If we would not throw it away, they will live happily ever after, I believe. This is a 3D structure. So as I mentioned, so we cultured them in suspension. Naturally, mammalian cells can adhere on the surface of the microalgae. And they can grow in aggregates like well distributed as a microcarrier, right? But thanks to natural properties of microalgae to produce oxygen, we don't have to think as much about oxygenizing the media. We just need to think more about the ratio of cells, of microalgae cells or mammalian cells. Naturally, the microalgae cells can divide in the same time as mammalian cells, but they usually divide 1 to 4. So they grow much faster, but not our microalgae cells, because we adapt them to 5% of light conditions, and they can divide 1 to 2, which is much easier for us to predict because they grow very the same as mammalian cells. Also, the large of aggregates can be much bigger in comparison just to monoculture, which is not shown in this picture, unfortunately, but this is simply because you have two biomasses at once. And also because the inner part of aggregate will not get to necrosis, so the cells are not dying from inside because it causes something which is called like angiogenesis-like effect. The media can still go through the aggregate. Oxygen is produced if you add light source to your bioreactor or incubator, whatever you have. We also checked the maintenance and expression of myogenic markers over muscle differentiation. I believe that we will not need this for later to differentiate into muscle, maybe just early myogenic markers to have presented for 3D structure or texturizing the process, but not necessarily for protein content as usually, because the nutrition comparison analysis we did, this is from public data available. It's, we just technically, if we will consider that we have a 50-50 percentage or ratio, 1-to-1 ratio of microalgae and mammalian cells in a hybrid product, you just averaging the nutritional parameters of each organisms, which we probably would not find a phytonutrients or diverse vitamins in a pork meat, but we will find this in microalgae. So that's why we call that our hybrid meat is fortified by microalgae. We also unveiled, not publicly, but for us, just this was not shown anywhere, so it's like you can see it. We made a Frankfurter. It's also 100% cell-based, but this is a reverse. So here is a majority of microalgae cells and 25% of porcine cells. It's still non-GMO. It's a true meat. It worked pretty well, but. So the benefits of a usage of microalgae in a system is that you still have a pork, because it's a hybrid biomass of pork cells and microalgae cells. We call this new pork because it's a fortified food product, hybrid product. It's longer lasting because microalgae have a naturally property to stabilize themselves against microbial contamination. Sorry. And higher availability means that you are able to scale in a higher yield in comparison to monoculture only. Yeah. We use, as I mentioned, we use microalgae to replace FBS. But the supplement we are using is not replacing the FBS. It gives the other advantages of that as well. So it very much depends what consortium of microalgae you are using, because there is 1,000 kinds of microalgae or species of microalgae on the planet. So and each has a nature property to do something, right? So if you want to have a better grow media, you will decide for one kind of microalgae, but the antimicrobial activity has a different kind. So you can do the consortia right according to your needs for media composition. We also produce growth factors using microalgae. So the microalgae are GMO in this moment, and they exudate the growth factors into media. So we use a microalgae to fortify the media. Then they are just filtered off, and we can use these media with the growth factors for our cells to feed them. It's cheaper because the media is done anyway for mammalian cells, and we can just transfer it from one corner of the lab to the other corner and culture it. We don't have to think as much about purity. We don't have to think about concentration, because once you can predict the yield of biomass of microalgae, what's the concentration of growth factors in your media then? We were awarded as a Mewery from United Nations for climate change, for SDG prize. And in the comparison study we do, we unveiled already 3 prototypes. We are on €8 per liter of media already. We are cutting down, but still it takes some time. So we're just 2 years on existing, so we have still some progress ahead, I believe. Yeah. And this is our last prototype. So we showed this on a rock festival in Czech Republic. The topic of the festival was sustainability, and we had a chance to speak in front of audience to present it, and you will be surprised, but everybody in the audience would be willing to have a bite of the burger. Yeah, and this is our Mewery team, so I will just most likely you know Roman, our founder and CEO. You know this guy, this is me. And this is a Delaware, he's sitting right there in the corner. And this is our CTO, and he's responsible as well for microalgae, because he's the head of the microalgae lab at Mendel University as well. So thank you.
Thank you so much. I'm pretty sure everyone here would also like a bite of your burger, so bring it next time.
No,
Yeah, questions there. So, yeah.
Nice talk. So, what flavor does the actual microalgae contribute to the pork?
It's a good question. Thank you. Everybody's interested in how it looks like if it's not green and how is the taste, if there are any off taste, right? That was the question. You just need to remove the color, and with the color you remove as well the off taste of the flavor of the algae. It's not presented in a different than red kinds of algae, so that's a solution.
So there's a question over there.
Hi. So if I understand correctly, you use the algae as a microcarrier, kind of. But what I see, my question is but the cells then they can only grow outside. Right? You don't have a porous surface where they can grow in, or do they?
The mammalian cells can adhere on the surface of microalgae, but they can also attach to each other in a suspension. So, because in the monoculture you can see also the aggregates of mammalian cells, right? So, but in a co-culture with microalgae, they are just like mixed together. Like there's no very much
Okay,
difference.
so you have a mixture of aggregates, kind of, with the microalgae and the cells themselves.
Yeah, it's a homogeneous population of algae and mammalian cells.
Okay.
So there's a question here. Yeah.
So you mentioned the scalability aspect. Many traditional biotechnologists like to avoid phototrophs because of light penetration issues. So how do you foresee the scale-up of these cultures?
How to avoid what exactly?
Because you're dealing with light penetration, so how do you foresee the scale-up? What kind of scales can you reach before light penetration becomes an issue? And how do you go bigger?
That's a good question. We are in a smaller scale currently, so we are not faced that now. But I believe that because we mixed in a final scale, we just mix the microalgae cells and mammalian cells. So we can mix them right in a. They could be dark if it's not for long. And anyway, the media is rich for nutrients. So then the microalgae cells do not require that much light because they can just eat some nutrients from the media. For example, they do not compete in any much nutrients, just the glucose. So you add twice as much glucose and they can grow together happily. And also the mammalian cells, they metabolize L-glutamine to ammonium, right? And it's toxic in certain concentrations. But microalgae can use ammonium and synthesize their own amino acids. So it's vice versa.
Right. Thank you.
A final question there.
Yeah.
So.
Lovely talk, and it's a really cool approach. I was just curious, umbilical cord stem cells I haven't seen around here too much, but they're pretty close to your standard MSCs. Are they in the same sort of proliferation range, if you're able to share?
Well, I'm not sure if I'm able to share, but I'm not sure if I'm willing to. I should share, but
okay, all right, no worries. Thank you.
Thank you.
All right, thank you then
Thank you very
again.
much.